He is joining.
Says joining.
Yes, sir.
Okay. Fine. So what we will do, we will try to understand what is counter current mechanism. I told you the very basic objective.
Yes,
The
sir.
basic objective is to make the urine concentrate, right? And so we know that the concentrated urine is actually present in case of all mammals, right? And it is the loop of Henle and vasa recta. Now, what is the vasa recta? Vasa recta is the series of the capillaries that is around the loop of Henle. Okay. So actually, it is not shown in the diagram. I can draw over here. So what happens that you see the loop of Henle, you can see over there, right? So in loop of Henle, what happens? There is a large number of the this pen gone. Yes, this is the pen. So what happened? Large number of the capillaries are around this, which I am drawn by the blue color. Okay.
Yes, sir.
So this is actually called as a vasa recta. So Henle loops and the vasa recta play a significant role in the counter current mechanism. So as the name suggests, counter current, that means that the two fluids is flowing in the two opposite direction, right? So in the Henle's loop, the fluid is flowing in opposite direction. So in the case of the vasa recta, okay. And since the fluid is flowing in two opposite direction, that helps in the very concentration of the fluid inside the filtrate. Okay, and how it is, we will discuss about that, right? But first of all, write down these 4 points. First of all, this is a very introductory of the counter current mechanism. Then one by one, we will discuss that exactly how it happens.
Yes, sir. Done, sir.
Okay.
So, Sheshi is trying to call me, but somehow I don't think she's able to hear me when I'm speaking. So I think it would be better if we could record actually for her.
No, actually, this session is actually already been in the process
Oh,
of
oh,
recording. Don't worry.
yes,
Okay.
sir. Yes, sir. Sure, thank you.
Sure. So let's talk about that how actually the counter current mechanism works. Okay. So first we need to understand the respective osmolarity. Okay, I think she is calling me. Hello. Hello. Shashi, join the session. What happened? Don't worry, actually I'm recording the session. I will share you with the recording. Okay. Okay, just keep trying because actually it is a network. Even your voice is actually breaking. So it means there is a network issue at your end, right? Oh, that is why, na? Okay, fine. So okay. So there's a network issue. I will okay. I will record the session. I will share the recording. Okay. Okay. Chalo. Okay, bye. So right, she was having some network issue. So let's try to understand the counter current mechanism. For this, we need to understand the osmolarity of the different parts of the kidney. Okay, and there are different figures you can see over there, right? So first, there is a upper region, what we call cortex region. You see the white portion, right? So that is a cortex region. So in case of cortex region, what you
So
see,
which one? Okay, yeah, the white portion. Yes, sir.
yeah, white portion is there, right? So this is the cortex. You can see the mouse now. I'm just moving the mouse. Is it visible to you?
Yes, sir. Yes, sir.
Okay. So this is a cortex, right? And this cortex, what you see, osmolarity is in the range of 300. You can see there, right? In case of medulla, as we go deep down, the osmolarity is going to increase. Or what you can see over there, that in the upper region, there is a 600. In the lower region, there is going to have a 900, 1200, and so on. Okay. Now, the question is that how the osmolarity changes and how this change of osmolarity is going to help in the concentration of the urine. Okay, so that is what we need to understand. So first, let's try to understand the exact role of loop of Henle. The filtrate is first of all going to enter into the PCT, that is proximal convoluted tubule, right? And from proximal convoluted tubule, it is entering into descending loop of Henle, right?
Yes, sir.
Now, in case of descending loop of Henle, the very feature of descending loop of Henle is that it is permeable to water, but not permeable to salt, right? So what happens over here, you can see in the diagram also by the blue color, the water is actually being coming out. Now, again, the question is that why the water is coming out? The reason is that at the outer region, the osmolarity is very high or the osmotic concentration is very high. And in the filtrate, the osmotic concentration is a low, right? So,
Yes,
and this
sir.
membrane is going to act as a semi-permeable membrane. And we know very well, due to osmosis, what will happen? Water will start flowing from the filtrate
Higher
into the interstitial
concentration.
fluid, right?
Yes, sir.
So it's very clear that when the filtrate will come down, for instance, we have started with the 300, when it will come down. So naturally, when the water will goes out, the osmolarity is going to increase, right? So as this comes down further, the osmolarity is going to increase from 600 to 900 to 1200 and so on,
Yes,
right?
sir.
Understood?
Yes, sir. Understood.
Right. So that is it is what is written over there. The proximity between Henle's loop and vasa recta as well as the counter current, maintaining the increasing osmolarity towards the inner medullary interstitium. Okay. So that means the
Yes, sir.
osmolarity is going to increase from 300 um, osmomole per liter to 1200. That is from the inner medulla. Okay. So please take a note of it. Then we will discuss about that what is going to happen in the ascending loop.
Yes, sir. Okay. Sir,
Yeah, tell me.
in the diagram it says passive and active, right? What does that mean?
Okay, so far, the movement of water from the descending loop to the interstitial fluid is going to be passive. Why? Because no energy is involved. Osmosis is a passive process because no ATP is involved, right?
Yes, sir.
So that is why it is written passive over there.
Okay, sir. So in the case of the active transport, there is ATP involved.
Right. In case of active,
Okay.
what happens? Since it has to be moved against the concentration gradient, so that's why ATP is required.
Okay, sir. Okay.
Noted?
Yes, sir. Okay.
Right. So what we'll see next, that what is the sequence of event that is going to happen in the descending limb? Oh, sorry, the ascending limb. So what happens that sodium chloride is transported from the ascending loop of the Henle loop to the descending loop of vasa recta. Now, first of all, let's try to understand the very nature of the ascending loop. The nature of the descending was what? They allows the passage of water, but they don't allow the passage of salt, right?
Yes,
Now,
sir.
ascending limb is, please remember, it is impermeable to water. They don't allow the passage of water. Okay, that is the ascending limb, right? But they allow the selective entry of or selective transport of salt or ions. Okay. So what happens? Sodium chloride is transported from the ascending loop of Henle to the descending loop of vasa recta. Right?
Yes, sir.
So why it is so? Because you see, let me draw the diagram for you. So you see, first we see that this is, let it be that this is a loop of Henle. Right. So this is a loop of Henle. Okay. And in this loop of Henle, what happens? There is another loop that is vasa recta is in around that, right? So that is the vasa recta.
Yes, sir. Yes, sir.
Now, what happens from here? The sodium chloride is moved out. And it moves to where? It moves to the blood. That is the
Yes, sir.
vasa recta. So it is sodium chloride is transported from the ascending loop of to the descending loop of vasa recta. Okay. So the ascending loop of vasa recta, what will happen? They will transport sodium chloride to interstitium. Anna, what is the interstitium?
Yes, sir.
The in-between area. This is what we call interstitium.
Yes, sir. Okay.
Right? So the ascending limb of vasa recta transport sodium chloride to interstitium. So when the sodium chloride is moved over here, what will happen? That osmolarity is going to increase. So what we have seen in the upper case, it was 300, right? But in the lower case, what happens? Even the interstitium liquid is going to have a osmolarity of 1200, isn't it?
Yes, sir.
Okay. So this situation is further created by transfer of urea. So from where urea comes from the, there are two places from where the urea comes. First, it is being transported by the descending loop of the loop of Henle to the interstitium. So the salt which I am talking about, that consists of sodium chloride as well as the urea. Okay.
Yes, sir.
At the same time, very small amount of urea is also transported from the collecting duct. Because you see, loop of Henle will goes to distal converted tubule, then it will go to the collecting duct, right? So from the collecting duct, very small amount of urea comes. So there are the two sources of urea. One, the descending loop of Henle to the interstitium. Okay.
Yes,
And
sir.
second, the collecting duct. Okay. So as a result, what will happen? As urine flows downwards in the collecting tubule, it encounters high and higher concentration of solute in the interstitium. So when there is going to have a higher concentration of interstitium, what will happen? It will go for more and more osmosis. And it will go for more
Yes, sir.
osmosis. That means there is a further concentration of what? The urine. Right?
Yes, sir. Yes, sir.
Please note it.
So,
Yeah, tell me.
we say like kidney stones, right? So then, what exactly? What exactly makes up the kidney stones?
Come
Like
again.
how? Uh,
You're talking about kidney stone, right? Is it?
yes, sir. Yes, sir. So, how exactly is
Actually,
it
you
formed?
see, kidney stone is nothing but it is some crystals of calcium.
Okay.
So what happens? They get deposited in the interstitial fluids. So
Okay, sir.
when their size become very big, what will happen? They will keep they will increase the pressure on what tubules. So tubule get
Yes,
constricted,
sir.
and thereby
Yes,
there
sir.
will be hindrance in the filtration. Okay.
Okay. Yes, sir. Yes, sir. Got it. Got it.
Done.
Yes, sir.
Okay. So we will discuss one more thing. What is juxtamedullary apparatus, right? And what is the
Yes, sir.
significance of the juxtamedullary apparatus? Okay. So first of all, it consists of juxtaglomerular cells and cells of macula densa. You can see there. So this is a juxtaglomerular cells. So mind it, these are the efferent arteriole, afferent arteriole, right? And in between them, there is a cell, what we call juxtaglomerular cell. Okay.
Yes, sir.
And also, there is going to have what? There is going to have this, the macula densa. So that cell is actually called as a macula densa. So both of them together is going to regulate the uh, sodium level in our body. So what happens?
Yes,
Suppose
sir.
there is a high sodium level. Okay. Then the macula densa cell trigger contraction of the afferent arteriole. So once the afferent arteriole is getting constricted, there will be reduced flow of blood to the glomerulus, and the glomerular filtration rate is going to be further decrease.
Yes,
Okay.
sir.
But just reverse is happening. Okay. Suppose the sodium level is low. Then in that case, what happens? Juxtaglomerular cells secrete renin with blood pressure when blood pressure in arteriole falls. So renin increases blood pressure via renin angiotensin aldosterone system. Okay. So this is how this entire regulation is basically exercised. Okay.
Yes, sir. Yes, sir. Done.
Done? Okay.
Yes, sir.
Then let's talk about that how actually kidney is actually being regulated. That is what we are going to study about that, right? Okay.
Yes, sir.
So there are three systems. What we call ADH, antidiuretic hormone. Then we have a renin, angiotensin, aldosterone system, right? And
Yes, sir.
then we also going to have atrial natriuretic factor. One by one, we will discuss about that. Please note it.
Yes, sir. Done.
Okay. So let's talk about first of all how actually antidiuretic factor acts. Okay. So here what we see that due to low blood volume, what happens? There is some osmoreceptors, right? And that osmoreceptors, what they do, they get activated and they release vasopressin or ADH, what we call antidiuretic hormone.
Yes, sir.
Right? And these antidiuretic hormone, okay, stimulate reabsorption of water from the distal part of the kidney, right?
Yes, sir.
So in case of sufficient body fluid, what happens? The osmoreceptors are switched off, okay, and what happens that once they are switched off, then ADH can also stimulate the constriction of blood vessels, resulting in increase in the blood pressure, right? Because initially there was a decrease in the blood pressure. So in this way, the antidiuretic hormone is going to act. Okay.
Yes, sir.
उसके बाद
Done, sir.
Okay. So we can see this flow chart. There is an ADH. So what will happen if there is going to have an increase in osmolarity of blood, right? What will happen? Hypothalamus is going to be excited through the osmoreceptors. So, and why this is going to happen? Because there is a increased osmolarity. That means the amount of water in blood is low. So this is also going to promote thirst. At the same time, it will excites the pituitary gland for the secretion of ADH. What ADH will do? ADH will tell kidney to reabsorb more and more water. So once the water will absorb, naturally water will go to the blood, and thereby the osmolarity is going to be reduced. Got it?
Yes, sir.
Please note it.
Yes, sir. Done.
Okay. Now there is a one more what we call renin angiotensin aldosterone system. So just we have seen the juxtaglomerular apparatus, right? So this is regulation being exercised by juxtaglomerular apparatus, and mechanism is popularly called as a renin angiotensin mechanism. So here what happens when the blood flow in the glomerular decreases, and when it will happen when there is a low blood pressure. So in that case, what will happen? Renin is released from juxtaglomerular cells. Now what renin will do? They will convert angiotensin in the blood to angiotensin 1 and further to angiotensin 2. So this angiotensin 2 is a powerful vasoconstrictor. And since it's a vasoconstrictor, what will happen? It will cause an increase in the glomerular filtration pressure and GFR. Because once the vessels is going to be constricted, the blood pressure is going to increase. And thereby, the glomerular blood pressure and the glomerular filtration rate is also going to increase. Okay. So this is a one act. But at the same time, it is also going to do one thing. And that is the angiotensin 2 also stimulate the release of aldosterone from adrenal cortex. And they facilitate the reabsorption of sodium ion and water from the distal part of the tubule. Okay. So as a result, this will also cause an increase in the glomerular blood pressure and GFR. So thereby, by twin action, what will happen? That the GFR rate of GFR is going to increase. Okay.
Yes, sir. So by twin action, what exactly do you mean?
Twin means, first of all, what angiotensin 2 was doing? It was constricting the vessels, so thereby increasing the blood pressure, right?
Yes, sir.
At the same time, it is also going to release the aldosterone from adrenal cortex, which
Yes,
eventually
sir.
helps
So
in the
happening simultaneously.
both axes.
Okay, okay, yes, sir. Both are similar and they do, I mean, they take place simultaneously.
Correct.
Yes, sir. Done, sir.
Done.
Yes, sir.
Okay. Then finally, there is a one more, what we call atrial natriuretic factor. So ANF is released when there is an increase in the blood flow in the atrium of the heart. You see, in the previous case, it was acting when there was decrease in the blood, but this is a case when there is an increase in the blood flow. So what it
Yes,
will
sir.
do, it will cause vasodilation. In the previous case, it was doing vasoconstriction, right? Here it is going for the vasodilation.
Dilation.
Understood,
Yes, sir.
right?
Yes, sir.
So the ANF mechanism provides the necessary check to the renin angiostensin mechanism. Okay.
Yes, sir. Done, sir. Sir?
Over.
Yes, sir.
So again, the flow chart, increase in blood pressure, atrial walls is going to release ANF, right? And the ANF inhibits renin release and thereby inhibit calcium secretions, right? So this is how it
Yes,
is
sir.
going to.
Okay.
Please note it.
Yes, sir. 1 second.
Noted.
Yes, sir. Last one. 1 second. Yes, sir. Done. Done.
Okay. Now there is a one, what we call maturation. Now, what is maturation? That is release of urine from the urinary bladder, what we call maturation, right? You know very well. Urine is stored in the urinary bladder and it will remain
Yes,
till a
sir.
voluntary signal is given by the CNS. Now,
Yes.
the signal is initiated by what? The stretching of the wall of the urinary bladder once it gets filled with the urine. So, in response to the stretch, what will happen? The walls of the bladder send signal to the CNS, and the CNS passes a motor message to initiate the contraction of the smooth muscles, and the bladder. Then simultaneously there is a relaxation of the urethral sphincter, causing the release of urine. Right. So the process of the release of urine, what we call it is a maturation. Understood?
Yes, sir.
So the urine formed, we know very well, light yellow colored, slightly acidic, and has a typical odor. On an average, 25 to 30 gram of urea is excreted per day. Right?
Yes, sir. Yes, sir.
Okay. So there are certain accessory excretory organ, So our lungs is also considered accessory excretory organ, right?
Yes, sir.
So because actually it removes CO2, that is a excretory product. That's why we call them accessory excretory organ. So is a liver. We know very well liver secretes bile, and that contains substances like bilirubin, biliverdin, cholesterol, degraded steroid, right?
Yes,
So
sir.
they eventually passes through the digestive waste. Now skin is can also be considered because the skin contains sweat gland which produce sweat, right? And they also release certain salts like sodium chloride, very small amount of urea, lactic acid, and so on. Okay.
Yes, sir.
So these are some of the accessory excretory organ. Please note it.
Written, sir.
Done.
Yes, sir.
Okay. So we know very well that sebaceous gland, that is primarily a microscopic exocrine gland,
Yes,
and
sir.
that also eliminates substances like sterols, hydrocarbons, right? And urea is also going to be excreted from there, right?
Sir, what are sterols?
Sterol is a type of steroids.
Yes, sir. Okay. Yes, sir. No.
Done.
Yes, sir.
Okay. So there are some renal disorders, or what we can say some sort of kidney disease, right? So what are the major kidney disease, what we know? So one is uremia, that malfunctioning of kidney results in the accumulation of urea in the blood. This is what we call uremia, right? Then we know very well that there is going to have uh, the renal calculi, that means some sort of stones,
Yes,
which
sir.
we are asking about. That
Yes, sir.
then we have a glomerulonephritis, that inflammation of the glomerular of the kidney. So you see, one of the parameters which is through which actually the very malfunction of the kidney is actually being noticed, and that is the creatinine. Okay. So whenever
Yes, sir.
there is a malfunctioning in the malfunctioning, then what happens? It is estimated with the creatinine level. Okay.
Yes, sir. Sir, which one did you say was similar to the? Yes, sir. Got it, got it. I'm sure. Yes, sir. Done.
Now finally, there is a one dialysis. So this is artificial kidney. So that is a machine that whenever there is going to have a failure of the kidney, right?
Yes, sir.
Then the compensation is actually done through that. Okay.
Yes, sir.
So how it performs? it performs the very functions of the kidney. Okay.
Yes, sir. Sir, only if dialysis doesn't work will a kidney transplant be done, right?
Sorry, come again.
Only if this doesn't work, a kidney transplant will be done.
Yeah. When there is a failure of the kidney, then only it works. Then actually, the patient has to be shifted to the dialysis, because you see what happens. The metabolic waste is actually being deposited in your body, and that kidney is the only organ that remove it. So naturally, the patient need to be
Yes, sir.
kept on the dialysis so that whatever the waste that is in the blood. So the dialysis is nothing but it is a process of filtering the blood.
Yes, sir. Yes, sir. Done.
ओके, चलो फिर आज के सेशन में हम लोगों को नहीं डिस्कस करना था. आई डोंट हैव टू डिस्कस एनीथिंग मोर, राइट? तो अगले सेशंस में वी विल टेक अ न्यू सिस्टम, फॉर इंस्टेंस लोकोमोशन एंड मूवमेंट. अगले क्लास में हम लोग डिस्कस करते हैं. ठीक है?
Okay, sir. Okay, sir. Thank you, sir. Good night.
चलो, थैंक यू.
Sir, do we have class on Saturday?
यस, ऑफ कोर्स, यस.
Okay, sir. Thank you. Good night, sir.
गुड नाइट.